Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Amelia Fitch about restoring microbial function in restored wetlands. Draining or restoring a wetland has a significant impact on the land's ability to store carbon. A lot of that has to do with the microbial communities that live there. But when a wetland is restored, how long does it really take for those communities to shift, and what does that mean for the land's carbon storage capacity? This episode, Amelia discusses her work studying wetland restoration and its effects on carbon sequestration and microbial communities. We'll talk more about that in a minute, but before we dive in, we want to thank our sponsor for this episode, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Amelia Fitch with us. Amelia grew up in coastal northwestern Oregon and first became interested in soil tromping around in restored wetlands as a seasonal park ranger. She completed her master's at the University of Cambridge and is finishing her dissertation on forest ecosystem ecology at Dartmouth College. Hi Amelia, how are you doing today?
Hi Abby, I'm doing well, thank you. I'm excited to be here.
Awesome. We are so excited to have you on and talk about one of my favorite ecosystems and one of yours, it seems, which is wetlands. So, you are working a lot with wetlands and carbon sequestration and restoration and all of those kinds of things. So could you just tell us a basic overview of specifically these coastal wetlands' role in things like carbon sequestration, how they sequester carbon, and just some of the history of the wetlands in the area and their relationship with that process?
Yeah, absolutely. So I think it will help if I tell a little bit of a background story on how I got interested in wetland restoration. So like you mentioned, I was working as a seasonal park ranger during the summers while I was attending University of Oregon, and I was helping out with some wetland restoration monitoring. So many years had gone by since these wetlands had been restored, and they had done a lot of planting of native plant species, and we were basically just tromping through them and seeing how the plant communities were responding. And it was super fun. It was incredibly muddy. Occasionally when the water would overtop our boots, we would just take our boots off and go barefoot because it was honestly easier. But anyway, so it was really exciting to be a part of that work, and I got to wondering about the sediment and what was happening kind of below the plant communities as wetlands were restored. So wetlands, beneath all those awesome plant communities that support things like, macroinvertebrates and birds, they're really important for carbon storage and for, providing nutrients that travel all the way up the food chain to things that we really, care about that are charismatic, like fish and birds. So you can think of them like a sponge. Like they're great for like protecting areas from storms when there's a lot of water influx from streams and rivers. They're like I said, they're super important for carbon storage. And one of the reasons why, which comes out in the research from this paper, is that they are anaerobic systems. And some of the listeners might know that anaerobic systems like wetlands also produce a lot of methane, but salt marshes that have a lot of salt in them, that have a tidal influence, they don't really produce methane. And we can talk about that a little bit more later. But these are like a very special type of wetland that is all over the world, but in coastal Oregon has been, these systems are recovering from being used for agricultural land for, a century or more. And so there's a lot of landowners and also groups that are interested in returning these wetlands because of these ecosystem services that are so important.
So obviously wetlands have like different layers to them. So you talked about how these were previously drained and then used for agricultural land. Now they're kind of letting the water go back in. So I'd like to know more about what that process looks like for both these tidal wetlands as well as these more freshwater ones in your region. Like, what are the layers? How do they come back together? What does restoring them look like?
It depends on the wetland system, and it also depends on what kind of resources are available. So if you have a lot of person power and you can plant a lot of native plants and excavate the historic tidal channels, then that might help, expedite getting that wetland back to what it looked like before. So these layers are, the sediment that can be meters deep of organic material, and some other minerals and bits of things, but it's really, really super organic. So when the wetlands are being drained for pasture use, often when there's a tide involved, you can build a dike and have a tide gate basically to allow or to prevent water from coming in. And there's often also with a dike that prevents water from coming in, a ditch around an area that you want to drain, basically. So it's on a trail when you would see an area dug off to the side to allow water to flow and keep the trail nice and dry and not muddy. That's kind of what they're doing, just on a huge scale. So when it comes to restoring those wetlands, for so long, the wetlands have been used for pasture. The soil is often very compacted, and if you let the water back in, it would start to add sediment layers on top of that compacted soil. But often there are tidal channels that used to go through a wetland and provide important habitat for salmon fry. And so one of the freshwater wetland that we looked at in this study, actually, they looked at historic photos of what the wetland used to look like from an aerial photo, and then they were able to recreate what those channels look like before moving on to the next layers. And then come in the plants, and the plants are also super important. They hold things in place, and they bring in more carbon from photosynthesis, and then, of course, all of the other habitat that they provide for other creatures.
Sure. I was just like, oh my gosh, they're doing time travel. This is kind of like turning back the clock. Sorry, nerd moment. So a lot of what you were looking at for this particular paper then was after these wetlands were drained and there was all the opportunity for that carbon to be released as it turned into the pasture land, and now you're looking to rebuild it. You were kind of focusing a lot specifically on, building up that organic matter in those layers, but also building up the microbial communities that used to live there. So can you tell me just, how were you going about testing that or encouraging that or just monitoring?
Yeah, totally. More of a testing or monitoring. I think doing some kind of inoculation of communities would be next level, and I would not be prepared for that. But yeah, so we were curious about, not only what is the carbon stock buildup in the accreted sediment. I just mean the sediment that gets slowly stacked on top of one another as the tide comes in, or as streams in a freshwater wetland bring in more carbon. There's actually a lot of carbon transported by streams and rivers and tides. Anyway, so we were interested in what the microbes were eating, and the way that we did this was by a couple different methods. So microbes, unlike us, they don't have stomachs. A lot of them are single-celled organisms. And so they have to do their digestion on the outsides of their cells. And what they do is they just spew these enzymes into the environment. And so a lot of people might be familiar with lactase and lactose intolerance. So if you lack the lactase enzyme, you can't break down lactose. So these enzymes that microbes are producing are similar. They're, targeting specific molecules. Some of them are more generalist, but anyway, so they produce these enzymes, and then the enzymes find molecules of organic matter out in the soil matrix, sometimes called the rhizosphere if it's around roots. And the enzymes cut specific bonds and break them into smaller bits and pieces that the microbes can actually take up into their cells and use as energy. So if you know the potential activity of these enzymes, you have a good idea of what is actually being broken down in the environment. And we looked at a bunch of different enzymes, and we got just like a kind of a broad brushstroke of across all of our sites, between the wetland that we called the reference site that had not been, diked or drained or disturbed in any way, between the reference site, the one that was restored, that had been disturbed and then restored, which is what I just said, and then the disturbed site. Across these different sites within the wetland complex, what do the enzyme profiles look like? And then we did a similar analysis looking at adding actual substrates of different kinds of stuff to the soil to see how the microbes would respire it. So we had, a couple of organic acids, some like amino acids, some, glucose and fructose, lignin, and all of these different things are just small molecules that have varying degrees of difficulty to break down. So glucose is a sugar, super easy to break down. As we all know, sugar, fast energy, compared to lignin, which if you look up a picture of the chemical structure of glucose compared to lignin, it's like an order of magnitude scarier for the lignin. It's like all different kinds of bonds going everywhere. And these require a lot of different enzymes to break down. So lignin is considered like a really difficult substrate. So adding all of these, mixing them up, adding little bits to our different soil samples, we were able to characterize by how much respiration came out of the soil when we added the substrates. What do these microbes like to eat? And with that profile, which we called a catabolic profile, catabolism, breaking stuff down, and with the enzyme profile, we were able to say, like, okay, these microbes are eating stuff that is, and decomposing, breaking down stuff that is similar or different, or, we were just able to compare across all our sites using this profile instead of comparing like individual substrates, if that makes sense.
Sure. It's kind of like if you put out a bunch of different food types, like in a forest, and then whatever they eat tells you what kind, birds ate all the seeds, so there must be birds here. And there's meat is gone, so there's probably, a wildcat or something, kind of like that.
Yeah, totally. Or if you had a bunch of different types of seeds that you put out and the creatures came out and ate, all the birch seeds, I don't know, cherry, whatever seeds there were. I feel like I'm getting down a rabbit hole here.
No, you're fine.
But yeah, then depending on the kind that they ate, you could say, oh yeah, there's probably an abundance of these seeds out there that they're used to eating. And so that would be the community that you would expect to be there.
Okay. So why is it that it is approached from that direction instead of, to continue the bird analogy, just going out with binoculars and finding them?
That's a great question. So I actually do a lot of DNA sequencing work now in my PhD, and with both of those experiences, they're getting at different questions. So if you're interested in who is there, then you would just sequence some DNA or maybe do some sort of like other test that would tell you the kinds of microbes that might be in the soil. But for our question, we were more interested in what was going on with the decomposition and like how the organic matter that was there was being processed. And we know that microbes are really important for that. We don't necessarily know who is there, but for our question, that didn't really matter. It was more about, for the microbes that are there, whether they're different, whether they're the same in the disturbed versus the restored site, how is their function different or the same? Because the function is the thing that's going to drive how decomposition is happening, and that could have effects on organic matter sticking around or going away.
Sure. Okay. So if you're trying to, this is like the opposite of the bird analogy, because I guess you're not trying to get rid of the carbon, but if you have like 5 birds that all eat sunflowers, you're like, I don't care who's eating them. I'm just glad there's no more sunflowers just hanging out in my bird feeder. That kind of, but like the opposite of that.
Yeah.
Cool. Okay. I'm so sorry. I just like completely derailed your research into birds.
I like the analogy, or like, okay, I'll take it one step further. So if you were thinking about a garden and you have a bunch of different vegetables growing, different kinds of flowers, different things that are available for animals to eat, and you want to know what is being targeted by something that might be eating your veggies, I would go out and survey like what is being eaten, and then I would, put up a fence to protect that specific vegetable rather than trying to see who is there. I guess I'm just going to poke a hole in my own analogy, but I guess you could also put traps out. But anyway, so that's kind of the thing that I was thinking about. Like, if you're worried about protecting something or knowing what is being eaten, then you would look at the process instead of the individual.
Everyone, I hope you're enjoying the show. Interested in learning more? Amelia's paper, Partial Recovery of Microbial Function in Restored Coastal Marshes of Oregon, USA, published in the Soil Science Society of America journal, will be freely available for the next 2 weeks. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thanks again also to our sponsor, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. Thank you for being our sponsor. Let's get back to the show. No, that makes a lot of sense. I think that's really helpful for me to understand. So then, can you tell me a little bit more about the process of these substrates? I mean, when you're first talking about it, I was like, I'm assuming you didn't just go out with your droppers and just put them in a marsh. So I would like to know, like, I'm assuming you're taking samples, and specifically because sediment is notoriously easy to move around, how were you able to get those samples? How much of it is water versus dusty little sediment particles versus that compacted soil? I would just love to know all about that process.
Yeah. Oh, great question. So between this was from my undergraduate research, my thesis, and between this work, my master's, and my entire 5 years of my PhD, this was the hardest field work and lab work by far. Huge. I have so much respect for wetland scientists and the people who choose to do this for their career, and maybe I'll get back to them one day. But holy cow. So starting out, you got to get your wetland walk down, because if you don't, you're going to lose a boot in the wetland or maybe fall over, and you have to have your PI pick you up out of the mud. And so once you're walking around in the wetland, we used like sections of PVC pipe, basically. Home Depot is a very excellent place to get research materials for this kind of work. And we would use this PVC pipe and probably a bread knife to like cut down through the roots. You would like use the knife to go around the PVC pipe as you slowly like kind of screwed it into the ground, trying not to compact the sediment as you went. And I remember getting just like circular cuts on my palm from doing this. I probably should have used gloves. I don't know why I wasn't, but just like trying to, it was very difficult to like screw them in and like pound them in. Anyway, once we had sediment in a PVC tube, we used rubber stoppers like underwater sometimes. Sometimes during the winter when we were sampling, there was so much water coming in that it was underwater. And we would cap them so that they stayed in their anaerobic condition. If it was the restored or reference site, the disturbed site was like super easy because it was a, cow pasture. And then getting them processed, we actually used an anaerobic chamber, which was super cool that Scott Bridgham had one of these in his lab. And we would basically like put any materials we needed in this little box, including our samples, and then it would flush all the oxygen out. And then you could open up the door on the inside of the chamber, and then you would use gloves that went into the chamber to like move stuff around. And I would sometimes forget, because we would be processing roots for like 12 hours straight. We were processing these samples to pick roots out. And I would sometimes forget that I was just gloved into this chamber looking in. And so one time I shouldn't be saying this. One time I sneezed and like went to cover my nose and mouth and just like sneezed all over the side of the anaerobic chamber because I was like gloved inside. This was pre-COVID though, so that was fine. And so getting the samples processed, they were, sometimes probably 80% water, 20% sediment. Maybe not that much, but sometimes it was like scraping sediment off of roots. And getting them into a place where we could then do these tests that I talk about, the catabolic profile, the enzymes, was also very interesting and only required a very tiny amount of soil. So for the catabolic profile, imagine you have a rectangular shaped plate and there's 96 holes. So it's like an 8x12. And we used little cards to scrape replicates of soil into these holes and get them to go down. And then we would add the tiny drops of the substrate to those holes too. So it was like a little microcosm of wetland sediment instead of like, doing it out in the wetland and measuring what happened out there.
Sure.
Yeah.
Oh, that's very cool. I love all of this. This is very exciting. How big are these tubes? The big rubber topper ones.
Yeah, I remember them being probably like 10, maybe 12 centimeters tall so that we had enough room to capture 10 centimeters of soil with the rubber stoppers on either end coming in about a centimeter. And the width was probably 4 centimeters.
Okay.
I'm just going on memory here.
Sure.
I'm like, this is a podcast, so you can't see what I'm doing, but I'm making a circle
Sure.
with my hands and looking at it. Yeah.
I'm just thinking about having to navigate through a wetland and gather just tubes and tubes of water. I mean, how many would you collect at a time?
We would get everything from a wetland complex in one day. So it was 30 of those tubes. And some of them were more soil than water, but yeah, sometimes they were underwater, but usually it was pretty manageable and they wouldn't leak or anything. Thankfully the rubber stoppers worked pretty well and they really didn't leak.
Yeah.
Yeah,
that was my other thought, is could quickly turn quite messy.
Yeah, no.
Well, that's wonderful. This is such a cool project. And so what did you find? What were your results?
Yeah, so I mentioned these wetland complexes. So we had one like saltwater tidally influenced marsh where we had, the 3 disturbed, reference, restored, and then we had the same 3 types of wetland sites, disturbed, reference, restored, in a freshwater site that not too far away. And the reason we wanted to include both is that, both are pretty prominent wetland types in this area and would likely could have different results because their plant communities are super different, like I talked about before, with like the saltwater ones needing to deal with salt. And they also had different wetland restoration tactics. So the freshwater marsh, they dug those tidal channels by looking at the time traveling photo, and then had native plant communities planted. And going to those sites, if I didn't know the history of those sites going there, I wouldn't have been able to tell the difference between that restored wetland and the reference wetland just by looking at it. However, once you dug down into the soil and you sampled the top 10 centimeters of accreted new stuff, and then the stuff below, it was very obvious that that 10 centimeters down sediment was compacted, had been ag land, because the properties were just really different. Like, bulk density is a term that we use to describe how thick the soil is. So you take into account the volume, you take into account the weight, and you know how much stuff is in that space. Super thick soil. And then the saltwater site, they basically just removed the dike and the tide gate. And so tidal influence was bringing sediment in, but it actually didn't have any plants growing on it yet, the restored site. The reference marsh did, a bunch of plants, mostly the stuff called pickleweed. But it hadn't been built up, the restored site hadn't built up enough sediment yet to really establish a plant community. So it would be interesting actually to go back now, since it's been, I don't know, almost, 10 years since I did this work? No, more like 7. And just to see what it looks like, because maybe plant communities are actually establishing. Yeah. And so an interesting thing that we found between these two sites, because of the different states of restoredness they were in, that the freshwater site actually had more similar microbial activity, more similar carbon stocks, and we didn't necessarily anticipate that, but we definitely thought that there would be something interesting to do with the salt influence, because once you introduce salt to the soil, the microbial community has to be different, or it has to be dominated by something different, because sulfate in a wetland, which is why it is stinky, is an important electron acceptor. We use oxygen as our electron acceptor, but sulfate is an important electron acceptor that can outcompete the methane producing microorganisms. That was a little bit jargony, but basically there's this battle going on for food in the soil, and depending on who has the more powerful electron acceptor that they can use will usually win. And sulfate beats out the methanogens. Another surprising thing we found was the soil carbon of that compacted below stuff that I was talking about. So in the restored site, sediment comes on top over time. The stuff below that used to be the ag pasture actually had a pretty high amount of carbon, and that's interesting. And of course, any grassland scientist listening is going to be duh, because grasses have a lot of root turnover, and they actually do add a lot of add a lot of carbon to the soil. And this is where it's important to think holistically about the history of these places and what was lost. And before, when I talked about those meters of really rich organic sediment being lost to that increase in microbial activity, that has to be taken into account. So, preventing any wetland degradation going forward is goal number 1, because we know what can happen when you dike and drain a wetland. Especially tidal wetland. And going forward with restoring wetlands, we also can see that it's going to take a long time, but that it is going to build back up the sediment that was lost and the organic matter that was lost.
Sure. So it seems like you've kind of moved on in your research focus, but I know that there were a lot, as there always is in science, more questions that could be asked. So where do you see, or maybe you're aware of research that has been done in the last 7 years that has asked some of these, but what are the next questions in future research? And then also, what's going on with you? What are you studying and moving towards?
Yeah, let's see. The next questions are what if I could study wetlands again in a perfect world? I think that it would be really interesting to apply the potential lessons learned from this study about the potential differences in the effects of restoration type, and just get out the word and try to collaborate with people who are are doing this and try to get some, not all of the data that we collected, but like it would be just so interesting to collect some of those indicators that we measured, whether it be like the catabolic profile, or at least just like looking at the carbon stocks of the soil, of the sediment, to see, if someone went ahead and planted native plant communities, what does that look like? Digging those historic tidal channels, what does that look like? We live in a place with a lot of wetlands, and I live in a place with a lot of wetlands, but there are very different places in the U.S. So like, what's going on there? Are these lessons applicable to other places? So that would be super cool, but also a huge undertaking. So maybe one day. Yeah. And then what am I doing? So I've been living in Vermont for the past 5 years doing a PhD, and I adopted a cattle dog named Waldo. And he comes out to the field with me. Love that guy. And so any kind of adventure that we can do outside together. I picked up mountain biking and generally just love being on a bike, and any kind of activity that Waldo can do with me is great.
Lovely. So then I have 2 final questions for you, because we snuck the fun facts in early, which I love.
Fun facts.
Yeah. So those are great fun facts. You can have more fun facts if you want me to ask you later. I can do that.
Okay. I keep them coming.
I love fun facts. So, okay. So then first of now 3, again, questions is if people want to learn more, where can they go?
Oh, yeah. Yeah. Okay. So, a couple things. If you are a landowner or you know somebody who has potential wetland on their property, there are possibly, I don't even really know Oregon very well, but there are possibly some tax incentives to maintaining and like protecting that wetland area. You can usually look up, let's see, for Oregon, it's called the Riparian Lands Tax Incentive Program. So I'm sure if you just Google whatever state you're in, like Riparian Lands Tax Incentive or something like that, you can probably look it up. My dad is a consulting forester, and he helps people work through that process. So there are people out there who can help you do that. There are also some very interesting articles about wetlands, and not only what they do for carbon storage, but just all kinds of different things, like clean water. So there's a science paper that hopefully can be included in the show notes or something that talks about that. And then just other things about wetlands, I think just being really aware of the stuff that goes down your drain is super important. So any kind of education you can give yourself or look up about things that are safe to use versus not safe to use, or better or worse. Probably not anything is perfect, but for example, sunscreen. Sunscreen is a huge one. Mineral sunscreen is a lot better than the other stuff, even though it feels kind of weird and cakey to put on and it's kind of annoying. It is a lot better for not just coral reefs, but a lot of different organisms that are out there in the riparian ecosystems.
Sure. So then we've got a couple suggestions already for what people can do to get involved, but if they want to get involved with wetland research or restoration even, what can they do?
Totally. I know of a few places in Oregon that would definitely welcome volunteers, and I'm sure that there are organizations all over that you can look up. One in Oregon where I grew up is called the Lower Columbia Estuarine Reserve, I believe. Estuarine Reserve. I actually did some volunteering with someone who was doing her PhD with them a while back, and we were looking at wetland plant communities again. It's called the Lower Columbia Estuary Partnership. So there's also another one called the South Slough Estuarine Research Preserve, and one of our sites was actually located within that research preserve. So I'm sure that there are all kinds of ways to be involved with those types of organizations.
Sure. So then, what is an additional fun fact that listeners wouldn't know about you if all they had was your research?
Additional fun fact, besides having a great dog, I really like to bake sourdough bread, and it's something that I picked up during the pandemic. So speaking of microbes, I learned that you can bake better bread if you follow a recipe really well. It's interesting being a lab scientist and then like getting home and like cooking or baking and being like, yeah, I don't need to follow a recipe. But it actually does really help. So I am an aspiring bread baker, but I'm still getting better and probably need to actually buy a food scale and a bread rising bowl.
Sure.
Next on the list.
Yeah. Baking is crazy specific. It's wild. Mm-hmm. So kudos to you.
I'm not a good baker. It's so funny. Like I'm so precise in lab, and then cup is already filled, and I can't fill it anymore on other parts of my life.
I get it. Well, that's a very fun fact. I mean, we all know Waldo is the most important fun fact, but the bonus fun fact is very nice. Well, this has been so great. Thank you for the work that you've done and are working towards in your PhD and into your future. So thank you so much for being on and taking the time.
Awesome. Yeah, no, thank you, Abby. This has been really fun. So I would also, I need to thank all of the people in the Bridgham lab who were there when I was doing my undergrad research. So Scott, of course, the lab PI, he took me in as an undergrad who wanted to do a research project on wetland restoration and helped me find sites. He came out with me to do field work. He was incredibly supportive for incredibly how busy he was. Lorian Reynolds was a graduate student when I was there, and she also took me under her wing and helped me troubleshoot all of these methods and helped me figure out data analysis, helped me write. Can't thank her enough. Kiana, who was my partner in crime on this whole thing, she's now working for the EPA, and yeah, we were doing field work together. We were doing lab work together. She did an incredible master's thesis using a lot of this work as well. And can't thank her enough. We spent a lot of late nights in the lab together trying to get all of those samples processed. And when we were finished, I remember going out to drinks to celebrate finishing the project. We decided that, I don't know if this should go in, but whatever, I'm going to say it anyway. We decided that if we had an artistic representation of our experience in the lab together, that it would be like a giant bucket of roots, and we would be like little figurines just slowly drowning into the roots, because that's how we felt.
Yeah.
Love
sure.
wetlands, but holy cow. Yeah. And then there were a couple of other folks, like Laurel Pfeiffermeister, who's now faculty member at University of Oregon, was also anytime I had a question, I would go to her. So such a supportive lab. It was really awesome.
Awesome. Well, that's wonderful. Thank you to all of those people that also helped with this research.
Yeah.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to S.K. Musfiq. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Hi, I'm S.K. Musfiqus Salihin, and my first name is S.K. Musfiq, and last name is Salihin. So I'm a PhD student at the Soil and Crop Science Department at Texas A&M University. I'm originally from Bangladesh, where I did my undergrad, and then I started my master's at New Mexico State University, finished my master's in 2020 spring, and then started my PhD in 2020 summer at Texas A&M.
Wonderful. And what are you currently researching?
So my current research is studying the greenhouse gas emissions and the soil health in organic cotton system using different cover crops, tillage, and biochar. So organic cotton is an emerging sector in worldwide, but US is lagging behind, especially the southern part or humid subtropical climate of US is not yet adapted to organic cotton. So here we are studying the adaptability of organic cotton in this part of Texas, in East Central Texas, and looking for the agroecosystem services with different cover crops, manure, and biochar.
Awesome. And if you could have your dream research project, what would that look like?
Nice. So my dream research would be to see the long-term effects of these organic practices, such as the manure, different cover crops, and biochar practices. What are the effects of these practices in long term, like at least 5 years or more? Because a lot of studies have been studying these for short term. They have a very good effect in the short term, like soil health benefits, agroecosystem services, but we still don't know how much they're going to affect or how much the impact is going to be in long term. So my dream project would be see the effects of these practices in long term.
Awesome. Well, if anyone would like to get in touch with SK Musfiq about his work, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you. Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.